think all of you have already seen the potential of ChatGPT or models like Stable Diffusion, which can generate images or DALL-E. So these are all different types of generative AI models. And fundamentally, these models work by predicting the next word, or in more precise terms, the next token. And I show you an example on the right here. For example, here, if I start with this sentence on my messaging app on my smartphone, I had such a, and the messaging app suggests great time. Or so this is like a very crude analogy of how generative AI works, which is how autocorrect works. So all these generative AI models, it is trained to predict what the next word in a series of words in a sentence, what the next word in a sentence should come, and as a result, is able to generate word after word after word after word based on the words it has already seen in the past. And that is how fundamentally how it works. But the interesting part is when you scale these models to billions of parameters, for example, ChatGPT, I think, which has 500 billion parameters, or Llama 2. So these models, something weird happens. These models develop the capability to reason. These models develop the capability to solve puzzles or write very fluent text. So this is what people have been observing, and people have been noting that when you are scaling these simple models, which at the basics, at the fundamental level, all it does is predict the next token or predict the next word. When you scale these models to billions of parameters, these models become really smart and really intelligent. And today, what I see around me is that leveraging these models in your day-to-day work has become absolutely essential, because these models can tremendously improve human productivity. There was a time, for example, when I used to write my code, and if I would encounter a bug, I would often go to websites like Stack Overflow or Reddit to find if people have already posted solutions to those bugs. But now you can just post just describe the bug to ChatGPT, and it comes up with solutions, just like that. And today I actually use software like Copilot, which actually helps you code faster. It actually helps. It helps in generating code, and sometimes really precise code, by the way. And what I have seen is that things that would take around, say, 2 to 3 days can done easily within an hour. So that is how it's boosting productivity, and it is going to become indispensable in the future. And this is true also for like other professions, people where they also have to write a lot of documentation, lawyers, for example, or even clinicians who often have to read a lot of patient notes to know what the patient is suffering from. So there's a lot of possibility now to use these tools by professionals in day-to-day life, which can tremendously accelerate the time to finish these jobs. So, but how can we use these tools as biological students or biomedical? If you're a biomedical researcher, how can you use these tools in your day-to-day life? And one of the most useful way is by using it to do your biomedical data analysis. Now, what I have observed during my time as a researcher, I often would work very closely with biomedical researchers and biomedical students. And one thing I have seen is that they would lack the necessary skills, or they would be limited by programming skills to implement the ideas that they have, or do the data analysis on the data that they have collected through their experiments. So back then there was no such solution, but today there is. So today you can actually use tools like you can use generative AI tools to actually do the biomedical data analysis, and it enables students and researchers without extensive coding skills to perform advanced data analysis and also visualizations. And you don't have to be super smart in coding. You just need to know some absolute basics. But all of these workflows, or all the analysis, can be done by statistics by using plain English. And we will show this today during our hands-on session, how to do this. So when you have tools like these to accelerate your data analysis, it then of course reduces the time and effort that would typically take, and so it frees up the time to focus on more high-level tasks such as designing your experiments or designing your hypothesis. So it tremendously improves the productivity of a biomedical researcher when you're using these tools in your day-to-day data analysis. And finally, it also offers opportunities to learn new things. Sometimes when you're learning a new algorithm, there's often a steep curve, but with generative AI tools, it can sometimes suggest new algorithms for your data analysis, and you can simply learn from it. And I think this is also a very good learning tool in a nutshell. So my my point is that biomedical professionals who master generative AI tools will lead the way in solving big challenges in biology and medicine, and I feel they will be irreplaceable. So let's talk a little bit about biomedical data analysis and what are the tools available right now. Of course, we have ChatGPT, which I'm sure all of you have used in your day-to-day life. But although it is free to use, one of the biggest disadvantages is that it results in a very inefficient programming workflow. For example, if you generate a code in the ChatGPT web app, so you are essentially copy pasting the code and copying the code and pasting it in a notebook somewhere. And as a result, your research becomes non-replicable or not reproducible. And there are also concerns with data confidentiality. For example, if you're working with very proprietary data sets, or so when you are putting this data on ChatGPT, on the ChatGPT web app, chances are that this data is seen by the ChatGPT model, and probably it's using this data to also fine-tune, also to improve itself. So this is an absolute no-no if you're working in a corporate environment where data security and privacy are taken very, very seriously. So that is why ChatGPT, the free version, is really frowned upon. Even I think in research environments, you should not use it when you are collecting very confidential data, where you're generating very confidential data from your experiments. You should not use the ChatGPT free app to your data analysis. So what are the other tools that we have? We have also Copilot, which is from GitHub or Microsoft. We also have a Copilot type system from Google as well, but these require, again, a monthly subscription of roughly around $10 to $20 a month. Although these tools fit seamlessly in your coding workflow, that is, you can easily generate code in your coding notebook. It often results in a very high cost, and of course, $10 to $20 per month is not affordable for most students and researchers. And finally, we also have the API tool. So I'll describe what API is during a hands-on session, so don't worry. So today Jupyter, I don't know if you have worked with Jupyter notebooks or Google Colab Python notebooks. There is a way to integrate the Jupyter AI, which is a type of Copilot functionality, but this Jupyter AI tool is powered by an API. Don't worry about what API is. It's powered by an API. And using this API is very, very cheap. It roughly costs around $0.0015 per 1,000 words, or roughly around 12 paisa per 750 words. And again, this results in a very excellent coding workflow. So it fits seamlessly in your notebook. It's very low cost. It's much lower than all the other tools that I have shown here, but it requires a little bit of manual setup. So, but in this session, I will also touch upon how to set these things up. So don't worry about it right now. And I'll also, I will not forget what an API is, because you will see what during the live demonstration. Cool. So what will you learn from this workshop? First, you will learn how to set up a Jupyter notebook using AI Copilot. You will learn a little bit of prompt engineering tips. You will understand what AI pair programming workflow is. So pair programming is a concept when in software engineering where you're essentially working with another person. And research has shown that when you work in tandem with one another, you tend to produce better code. And this is also true for when you're working with AI to generate code. So I'll show you what pair programming workflow is. I'll also show you a little bit of on the basics of Git, which is really important if you want to share your research with other people, and it also allows one to do reproducible research. And finally, I'll talk a little bit about data confidentiality as well. All right. So now I will hand over the presentation to Alok, who will describe the case study that we have chosen for this workshop. And this case study, then we will do a coding walkthrough where I'll explain various concepts that I just discussed before. So, but to explain the case study, I will need Alok's help.
Yeah,
Alok,
sure.
over to you.
Yeah, hi guys. So guys, like as Neel basically like introduced you about the generative AI and its application. So today in this case study, basically, we are going to look at one of the small data set. And what is the data set that I've explained you in detail, basically in the last class and before that, how do the data sets look like, right? So today in this hands-on, we're going to take a small data set from CBio portal. So CBio portal is basically a website or of a database like where you can find a huge amount of the data sets in the field of cancer, first of all. Okay. From there, we have taken this pediatric brain cancer data set. Okay. And this data set is basically containing sample size for the 218 patients. Okay. And as it is a data of the brain cancer, so we know like the brain cancer again varies for the different stages. Okay. So there are different stages of brain cancer. So from there, like this particular data set, the 218 patients are stratified or present of the different categories following under medulloblastoma, ganglioglioma, LGG, which is for low-grade glioma, ependydoma, high-grade glioma, craniopharyngioma, and atypical teratoid rhabdoid tumors. So these are all the different forms of the tumor of the brain tumors, basically. Okay. For the demonstration purpose in this session, we have only taken two categories or the samples from the two categories here. MBL, which stands for medulloblastoma, and GNG, which stands for ganglioma. Okay, and for the sake of explanation, we have only taken 2,000 genes from the approx 22,000 genes. Okay, so the data sets upon this particular website contains approx 23,000 genes. From there, we have taken only 2,000 genes and samples coming from the MBL and the GNG classes. Yeah, Neel, can you please go next slide? Great. Now, so now we should need to understand like what do we understand by GNG and the MBL. So as I told you, like these are the two different types of the brain tumor. So medulloblastoma is one of the CNS central nervous system embryonal tumors arising from the cerebellum. So we know that like the brain is divided into the different segments and it occurs basically from the cerebellum. And it is one of the most common pediatric malignant brain tumor. Okay. And it comprises approximately 50% of all the pediatric CNS tumors. And about the GNG, it can occur anywhere in the CNS. So it can take place either in the spinal cord as well, or in the cerebrum, or other parts of the central nervous system. Okay, and are most commonly found in the temporal lobe of the brain. So this is basically about the data description. Okay, yeah, Neel, we can go next. Great. Now, so in this hands-on session, what we are going to do basically is that the data set that we have taken. Now we know that there will be approximately 34 samples of the two classes, and there will be 2,000 genes, and one final variable will be there, which specifies the patient belongs to which class, and the class here stands for GNG and the MBL. So right now our task will be basically to perform and find out the differentially expressed set of the genes. What do we mean by differentially expressed set of the genes? We have already explained in the like yesterday's class and the day before that class, right? So now you understand that. But here we'll be finding those using very simple t-test between the two classes. Okay, so we are not going to make use of any complex models, but we will simply make use of t-test. Now the point is, after finding out the genes, now in the next question will be how do the samples look like? Okay, so for that we'll be performing basically PCA. Okay, now we'll be performing PCA to see how the samples are present with respect to each other upon the basis of these set of the genes, which will be existing as the differentially expressed. And after that, we'll also be interested to know what are the different pathways in which the genes are enriched that we can perform as well. Okay, to look for the pathways in which the genes are enriched. And in the end, we'll try to see the genes that we have been able to get, how the samples are being classified based upon this set of the genes into the GNG class and the MBL class. So this is the wholesome idea of this particular hands-on session. And we'll be doing all this thing with the help of generative AI, where you will see that you really need to have good understanding of all these technical things. So solid understanding of the domain understanding of all these things is required. But if you don't know have good programming skills, that is fine. But you need to understand a little bit of programming, right, Neel?
Absolutely, and you will see the reason why.
Yes, and so now, so yeah, so guys, till here, if anyone have any doubts, feel free to ask us, right, Neel?
Yes.
Yes.
I hope the case study is clear to you. So we can take questions on the case study if you have before we can start the coding walkthrough.
Yes. So we can have the question for 1 to 2 minutes. If anyone have any questions, please go ahead. Hello. Guys, do you have any questions? Or shall we go ahead?
Sir, there is no question, but can you please go back to the case study slide?
Okay. Neel, case study, yes. Yes. हाँ जी बताओ तान्या.
सर मेरा क्लासेस रह गया था देखने के लिए तो बस वही देखना था.
Okay. So we are going to take here two classes, medulloblastoma and ganglioglioma. Okay. Yeah. So I think Neel, then we can start.
Okay, so let's start with the coding session. So first of all, this setup, I would urge all of you to scan this QR code, and this will give you a link to our GitHub repository where you will find the entire code. So please do this right now, and you will see all the instructions. You'll find all the instructions on how to set up the whole coding environment. But in a nutshell, we are using three main things. One is Jupyter Lab, which is an open source Python software. Then we will use some packages such as Jupyter AI, OpenAI. I'm sure you have heard of what OpenAI is, which the company behind ChatGPT, and they have a Python package to use these models programmatically, which is called the OpenAI Python package. And we have we also need the OpenAI API keys. So as I promised to you, I'll explain what API keys are. So API keys are it's a special piece of, it's a password. Think of it as a password, which you need to access the ChatGPT models in a programmatic way. Of course, OpenAI does not want everyone to access these models for free. So you will need to first set up an account on the OpenAI website. And they also need a credit card for this. That's unfortunately the con that they need a credit card, because they will charge you based on the number of times, based on the number of words you have processed using their OpenAI models. But as I showed you, the cost was absolutely minimal, 12 paisa for roughly 750 words, which is roughly, I think, 2-3 pages of A4 text, which is almost nothing. But essentially, what these keys are, they're kind of a password which you need to access these models in a programmatic way. And just to quickly show you, if you have scanned this code, you would have arrived at this repository, which provides a lot more information on what the case study is, the objectives are, and there are also instructions on how to set this up. Now, we have used the advanced instructions to set it up on a local machine, simply because when we were testing out the Jupyter AI functionality, which you will see how we are going to use, it was working much better on our laptops locally than when we tested it on Google Colab. There were some bugs, and I think they will fix it in the near future. So for the purpose of this presentation, we are using the advanced setup on our local machine, but this can easily be done on Google Colab. And I have already provided some instructions on how to a way around some of these bugs that you will see on Google when you use Google Colab. So please feel free to go through these instructions whenever you would like to try this out yourself. Okay, so now let's actually go to our coding notebook. So this is the Jupyter Lab, as I mentioned, which is an open source Python software. Python, actually, it's called DE, Integrated Development Environment. And this runs on your browser, for those of you who do not know. So if you are using Google Colab, you do not have to do anything except for executing this first step line. Now I have commented this line because I have done it in a manual way. So these things were done in a command terminal. Don't worry about it. But if you're using Google Colab, so I'll show you. I don't know if you, Alok, if you have shown if you have used Google Colab in this presentation. But
yes. So I have shown them Google Colab. Yes,
okay.
So they know what is Google Colab basically.
Okay, then it's good. Then all you need to do, if you're using Google Colab, you just need to upload this notebook from the repository and just install these packages which I mentioned here. That's all you need to know. That's all you need to do to get started. Okay, so let's start with first by configuring the Open API keys. So here I will first need to provide these special passwords so that I can use the OpenAI models in a programmatic way. So this is this block of code I'll just execute, and it asks me to enter my OpenAI key. These keys I have already copy pasted I have already saved on a local file. So I will just copy paste them. But wait, I can already show you where I have. So you need to go to this particular link to get this OpenAI keys. So I'll just show you, quickly what this looks like. Okay, login. Just Google. And here you can see my secret key here. And I have copy paste. I have already saved this key on a local text file. So I'm going to just use this key in the workbook. So going back to the workbook again, I'm gonna paste here and press enter. And this has now accepted the password to access these models. The next thing that we will use now is to set up the Jupyter Copilot. So this Copilot is essentially the AI that you will work with during this coding walkthrough. And this is called Jupyter AI. So I will just load this library first. It's not a library, it's called a magic function. So this has loaded. So, okay, so now I will demonstrate how we will be using generative AI, or how we will use models like ChatGPT to do our analysis. So the first things first that we need to do is write a magic command. So here, this percentage is the start of the magic command here, followed by AI. Then I mentioned which model I want to use. Now this Jupyter AI tool, it supports various models, but in this particular coding walkthrough, we will be using the OpenAI model, for which I have already entered the keys. So OpenAI, I describe which model I want to use. So OpenAI chat, I want to use this particular model, GPT 3.5 Turbo. There are also many other models, such as the latest ones are GPT-4 and GPT-4 Turbo. But for the purpose of this exercise, we'll keep ourselves limited to GPT 3.5 Turbo. So this is the magic command line. And following this line, I can just give an instruction or a prompt, which is say, write a short poem about ChatGPT. And I'm just gonna go ahead and execute this line, execute this block of code. Okay, and there you see, this has come up with a very interesting poem. You can read it in your free time, but this is what in a nutshell it does. You give the magic command, you write your instruction, and voila, you get the output from this model. So let's do another warm-up exercise. So again, we write the magic command here, but this time we add an additional description. It's called a flag. We add the -f code flag, which means I want the output only as a code. I don't want this verbose stuff. I just need only the code output. And this is my instruction now. This is my prompt: generate a function to add two numbers, then print out the sum of 2 and 3. This was previously done. I'll just delete this just to show you what will happen if I execute this block of code. There I go. And there you see AI generated code inserted below, and it has immediately given me this code definition: add two numbers A and B, and it returns the sum of A and B. And if I can just execute this, you can see sum of 2 and 3 as 5. We can also generate visualizations. We can generate code to generate plots. Again, the same funda. We just add the magic command here first and add the format code flag. -f and -- format code, they're one and the same thing. So in this particular case, I want to create a function that plots a circle of radius r, and let's plot the circle of radius r equal to 4. And there I go. There we go. We have the code here. It uses the matplotlib library. And let's see what happens if we put this. And you can see it has generated the plot for a circle of radius 4. Any questions on this? Any doubt? Did you understand? So this was just a warm-up exercise we did. We will now actually get into the nitty-gritty of the case study that Alok had described. So before we proceed with the case study, let me give you a brief primer on what prompt engineering is. So this, so what essentially we were doing was we were giving prompts and we were getting outputs. So creating prompts to get generative AI models to produce precise outputs is called actually prompt engineering. So the more instructive your prompt is, the more precise your prompt is, the better output you will get from the model. It's the same funda as garbage in or garbage out. Or if you have good input, then you will probably get a good output. So you have to really make the prompts very precise. And we have a few tips on how to do this, how to make the prompts precise so that you can get very specific outputs. And here are these tips. So keep the prompts limited to one data analysis task. For example, it is not recommended that you instruct the model to do a t-test, a PC analysis, and a machine learning analysis in the same prompt. Probably it's not a good idea. Instead, you should keep the whole prompt limited to one data analysis task whenever possible. You also have to define clearly what the input data set is for the data analysis task. For example, if you are telling the model, okay, I have a data frame, you have to define what the columns in this data frame mean before instructing it to do a very specific analysis. So please define what the inputs would be. At the same time, it's also very important that what the output should be. You should clearly define what the output should be. For example, whether the output is just a number, just a piece of string or a text, or whether it should be a plot. And it is always a good idea to wrap your code inside Python functions. What I mean by that is you should create an instruction like this: give me a Python function to do the sum of A and B, instead of write a code to sum up A and B. So you should always use functions to wrap your code. The reason being, functions are reusable. You can share these functions with your colleagues, or you can reuse these functions at a later stage. And it's always a good idea to keep your code as much reusable as possible. And finally, just like humans, the AI will never know what's in your mind in the first go. So you'll always have to iterate, and you will see why it is. You will see during our live demo. So you will need to have some very basics of how or what a Python code is. So you should know a little bit on the basics so that you can debug the code. The AI will generate a decent code in the first go, but there will always be some very specific. There will be some mistakes which you need to figure out and then correct it, and so that you can execute it easily. That is why the knowledge of coding as well as some patience, and you should be ready to iterate as much as possible. And these are the 5 tips, prompt engineering things that you should know to do data analysis in an effective way. Having said that, let's now jump back to analysis. So we will first load the data set now. And this is the data set that Alok had described, which is gene expression, data set. And you'll see I have printed the top 5 rows of this data set. I think these are gene expression counts. Alok, am I correct?
Absolutely, yes, right.
Yes, so these are gene expression counts. These are various genes. So the first analysis I think that we should try now is whether differentially expressed genes. We should try to we'll use t-tests to find differentially expressed genes.
Yeah, Neel, we have a question here. Yeah.
Sir, I was asking about creating a circle. Did it created a circle or gave a code which on applying generated a circle?
Yeah, it gave a code which on applying generated a circle.
Oh, okay. Thank you.
Any other questions?
Yes, there was one more question in the chat. So I answered, but that will be great. Can't we just import? Oh, fine. So I've answered that question only. Okay, fine. Can't we just import maths? Oh, fine. So this was the question. It is, can't we just import maths for that circle problem? So they're basically trying to ask like, Tanya, can you ask the question like more prompt exactly? Like what do you want to ask? Tanya, are you there?
यस सर सर वो वही था कि हम पूरा
क्या?
वो मैनुअली कोड टाइप कर सकते हैं मैथ्स को इंपोर्ट करके और उसके बाद विजुअलाइज कर सकते हैं सर्कल को, पर वो खुद ही बना के दे रहा है, एआई जनरेटर है हेल्प करने के लिए, वो आपने आंसर कर दिया.
राइट. बट अभी आप ये देखोगे जो एआई जो आंसर देगा वो रॉन्ग आंसर्स भी देगा बहुत सारे, ठीक है, तो वहां पे फिर आपकी प्रोग्रामिंग अंडरस्टैंडिंग आएगी.
इसमें कोई प्रॉब्लम नहीं थी. You will see that AI will often make a mistake when the prompt. This is circle. The circle code is very well known, so
yes,
no problem will happen with this. But let's go some more interesting examples, which is related to our case study. तभी आपको पता चलेगा कि कितने mistakes होती हैं AI से.
absolutely.
So for example, now we want to use do this analysis of we want to find out differential expressed genes using t-tests between the 2 clinical conditions. So I think if you look in this data set, there are 2 clinical conditions. One is GNG and the other one was what? I forgot the name exactly.
MBL, medulloblastoma.
Yeah, okay. So we have 2 conditions. So we need to find out whether the gene expression is different or not based on the t-test. And for this, again, we'll first use the magic command. And now we have already prepared the prompts here. So you are a bioinformatician having
So
experience
Neel,
with
I'd like
Python.
to add here one thing. Neel, just a minute, sorry.
Uh,
Yeah. So Neel, just to make the session a little more interactive, I think like what we should do. So we should like hide the prompt and let the student ask कि भाई आप इसे कैसे करोगे problem solve पहले.
हाँ, that's a good idea. मैं इसको ऊपर
Right.
थोड़ा scroll up कर देता हूँ. And
Yes.
you guys can like suggest prompts and then I will try these prompts to execute it.
Right. Yeah, hi, Payal, are you there? Hello. Hello, Payal, are you there? I guess she's not there right now. Harshvin, are you there? Hello.
Yes, sir.
हाँ जी, आप बताइए. So like day before yesterday class, when I was teaching you biostats in detail, so मैंने आपको proper like t-test समझाया था detail में, am I right?
Yes, sir.
चलो good. So we know that what is the t-test. That is like we are given two different distribution and we want to compare their means, right? Now अब हमें ये करना है basically हर्षिन कि now we want to perform the t-test upon all the data to get the result. So अब आप ये करोगे हमें यहाँ पे manually कैसे काम करना चाहिए? You are not given any AI. So अब आप ये काम कैसे करोगे? You have to instruct us. Just think the problem, Harshit. क्या करना चाहिए हमें? Is the problem clear to you, Harshit?
No, sir. Can you repeat?
हमें ये करना है. We need to perform the t-test to find out a particular gene is differentially expressed or not.
Yes, sir.
Okay. What are the two classes? And what are the two classes? GNG and the MBL. So how are we going to ask this question to somebody?
Alok, can I ask?
Yeah, please go ahead.
Here you have selected only one class, so how can it be
हमने
compared
select
with
नहीं किया. हमने select नहीं किया. वो
Okay.
डेटा के अंदर पहली 5 रो में GNG सैंपल्स थे ओनली, इसलिए वो खाली GNG दिख के आ रहा है. ठीक है. हमने खाली डेटा को रीड करके हेड प्रिंट किया. वो डेटा के अंदर MBL क्लासेस हैं. Got it?
Okay.
Yeah. So Harshit, now you have to instruct. बताओ कैसे करोगे?
Sir, I don't remember how to run the t-test.
चलो डिटेल्स रख के सब छोड़ दो. Yes, somebody was saying something.
सर पेयर t-test रन करके p वैल्यू निकाल के फिर नेविगेट करेंगे आगे.
चलो that's good. But paired नहीं, pair wise होगा. ठीक है. Paired is different. Pair wise होगा. ठीक है. Right. So तन्ने कैसे करोगे? Like pair wise आप कैसे करोगे? एक जीन पे करोगे, सब पे करोगे, कैसे करोगे? Just try to think. कैसे करोगे आप?
सर दोनों सैंपल को अलग अलग ग्रुप में रख के और GNG और MBL के जो सैंपल्स है और फिर पेयर में बना के करेंगे पर
So,
जीन की.
Absolutely right. So right, she's right, Neel?
Yeah.
Great. So good, you are going on the right direction. That is like yes. Vaibhav, would you like to add something here?
No, sir.
Good. So Neel, now we can then go with the prompt.
I think Shwetamri, you have a question on one.
No, I have asked. Sorry,
Okay, great. Yeah. Okay, so let me tell you guys the problem कैसे solve होगा. So we need to perform t-test upon each gene with the two classes. So as Tanya said, that we need to first filter out the data based on the two classes and then take each gene and perform the t-test. Very simple, right? But we need to now specify this question very specifically. क्योंकि आप जैसा आप input दो वैसा आपको output मिलने वाला है, right? So let us now see that how are you going to design this prompt and ask the question to the chat, like this is not ChatGPT, but to the AI, like, and how it is going to answer us. So let us see for that. Yeah, Neel, we can go ahead.
Yeah, for that we already prepared a prompt, but you can also suggest your prompts, to do the same thing. But the prompt that we designed was, you are a bioinformatician having experience with Python programming language. You are given a gene expression data. Again, we are defining the inputs now. A gene expression data with each row is a sample and each column is a gene expression value. The last column is clinical status. So this is the last column here. So here, this is a random sample that I have now shown that you can see both these statuses, GNG and MBL. So the last column is a clinical status, which is a categorical variable with classes GNG and MBL. So design a function. Again, we are asking you to wrap the code in a Python function to perform t-test between the two classes for each gene and return the results as a data frame with t statistic and p-value in columns. Are you satisfied? Like, do you want to anyone want to suggest anything else for this?
Right.
Do you think the grammar is wrong or something? We can make some
Yes.
correction.
So Tanya, now do you agree? Like job में कहा था कुछ ऐसे कहा था?
yes,
sir.
it's fine.
Good. तो इस टाइप से प्रॉब्लम को डिफाइन करेंगे, ठीक है? एंड यू कैन सी हमने पूरा एक इन्फॉर्मेशन जितना मैक्सिमम दे सकते हैं तो मैक्सिमम इन्फॉर्मेशन को दिया है.
Okay,
So now
cool.
let us
Chalo,
see.
I will, let's see what will happen. So I'm now executing it. And the AI has given me this code. So let's see step by step what it has done. Gene expression, first it has taken all the data until the last column, and the clinical status is put in the last, it's taken from the last column, I think.
Yes.
Has created a data frame. I think this mistake, there is a mistake here. I'm sure I'll show you what this mistake is. I think I know what it is. But the logic is correct. In the logic is, in a nutshell, it is correct, because what it does is it goes for each gene in the column. It creates two groups, group 1 GNG, group 2 MBL. I think Tanya, you correctly mentioned that. And then it just uses if scipy.stat t-test in function to calculate the t-statistic and p-value. And then it adds the results in a results data frame. Now there's a technical error here, and you will see what the technical error is. Let me see now what is being run. So I don't need to rerun this particular line because I have already loaded this data set here, workshop data.csv, but it has suggested some new name, but we don't need this. So I just comment it out. T-test, perform t-test, gene expression data. Actually, the name was gene exp. So I will just put here the gene exp. Sorry, here it actually takes data. So I will do what I'll do is I'll data equal gene exp, and let's see what will happen. Again, the error here is that I think this error we encounter every time whenever we run this. It's a technical error. What it means simply that data frame, does not have this function append. I think this is a list. This is a function that only exists for lists, where you're appending. If you know about Python programming, so usually we add items in a list, but in a data frame you don't have that functionality. So go ahead.
सर जब हम R में काम करेंगे तो जो हमारा डेटा सेट्स होता है वो इम्यूटेबल ही रहेगा.
Okay.
We can't make changes in it.
No, we are not making changes in this data set. The original data set immutable ही रहेगा. We are not making changes to the original data set. That's why हमने यहाँ पे एक अलग से data frame declare किया है, results df, and we are just calculating and saving the results in this new data set.
Okay.
So we have already debugged this code for this particular exercise, and this was in a different notebook called Workshop Codebook Final. You will find it in our repository. And we actually corrected this code instead of using the results, instead of declaring an empty data frame with these columns, we actually used a Karthik, I'll just come to a second. We actually used the list object, and we calculated all these differences and saved it in a list object. And you can see, so results is an empty list. We create the two groups. We calculated the static p value. We calculate the I think the log change value as well. But this is a different. Wait, I think this. Sorry, this one.
Yes.
Yeah. So here we actually calculate. So this is the first exercise. So we declared an empty list. We did all the calculation for each gene separated by the clinical condition. We added these results in these two empty lists, and using these two empty lists, at the end we generated the data frame. So this code will run for sure because we have debugged this. So let's just to save some time right now. We'll just use this code, and you will see that this will run. And there you go. If I just look at the head, I see 5 genes, and I get the t statistic, and I get the p value. All right. So
You go on hold.
Can you come again? Sorry, hello.
Sorry, Neel, you had just got stuck there for some time.
Oh, okay. So I hope you can see my screen, the results, and this code. If anyone of you have any question on what this code is doing, this is very close to what Tanya was mentioning. So yeah, we just simply did a technical change in the code that was generated by generative AI. And that is why you should know how to debug this code. Sometimes you should have some fundamental understanding of different types of data structures. But nonetheless, this is the code that you have now, and you can see the results, the t statistic and p value calculated to see whether significant differences exist for each gene between the two conditions or not.
So guys,
Okay,
anyone
what
need
is
doubts?
SciPy stats? What Alok, sorry?
No, sorry, I'm just asking any doubts.
Now, we have a question. What is SciPy stats? So SciPy stats is a library with which you can do a lot of statistical analysis in Python.
So Alok, p-value should be less than 0.05.
It depends upon you, like what threshold you're going to select. It depends upon that. You can choose the threshold for 0.05. You can take the threshold at 0.01 or 1e minus 10. It depends upon you, basically. Okay.
Okay.
Yeah. Fine. Great. So any other
Okay,
question,
so-
guys? Tanya, is it clear now to you?
Yes, sir.
Hello, good. Yeah. So yeah. So now you can go to the next.
Okay, so now we'll ask the model, we'll instruct it to update the previous function, this function, to calculate the log fold change values for these two clinical conditions. But you will see there will be an error here. So now what has happened is it did not update this function, although our instruction was update this particular function. What it means is that this tool does not have memory of what has happened in the past. So you should define each instruction from the very beginning. So each instruction should be complete in itself. So to do the same analysis, what we need to do is now define the prompt in its entirety. So again, what we did was UI bioinformatics, you are given a gene expression data where each is a sample and each column is a gene expression value. We define what these inputs are. Okay, so now this is what we added. Design a function to perform t-test and the log fold change between the two classes for each chain and return the results as a data frame with t-statistic and p-value in columns. And let's keep our fingers crossed and see if this code comes out correct or not. Okay, so what it did was, so it again, so this function looks very similar. It defined some empty lists, then it looped over, it did a for loop over each chain, calculated the t-statistic and the p-values, and also calculated the log fold change. And at the end, using these three lists, it created a data frame, and then it printed and returned the data frame. I think this should work. I have a feeling that.
Yeah. Neel, but there is a mistake here in the code, basically. There is one mistake here in the code, and that is not technical mistake. That is a conceptual mistake here. That is not mistake, basically. That is not mistake. But the model is like, but the prompt is not understanding. Go like, we have not told it the data was either normalized or not, right? So basically, what we can see here, guys, is that at this step of log fold change calculation in the model, in the for loop, where we are calculating the log underscore fold underscore change dot append. So because the data is already log normalized, log transformed, okay. So when the data is log transformed, so if you have to divide, then it will go to the minus. But we know that log of A by B becomes log A minus log B. Okay, guys. So if the data is normalized, that you should always subtract the mean value of the two different features. But if the data would have not been normalized, that would have divided. Okay. And why this problem came? Because आपने model को, आपने prompt को नहीं बताया that your data is either normalized or not. If you would have told that your data is log normalized as well, then you would have definitely created a minus sign there, most probably. Okay, guys. So you should know this thing. If you know a little bit of logarithm, so log of A upon B is equal to log A minus log B. So we should not Neel basically like replace that divide by minus.
Here, right? Minus.
Like आगे code में where we are dividing the mean. Yes. In this line only, yes. This divide by mean. Minus. Divided जाएगा minus आ जाएगा. Yes. And replace the divide sign. Replace the divide.
Replace the divide by minus. Okay.
Yes. Now it is right. Yes. Replace. Yes.
Okay.
Now it is right.
Okay.
Yeah, Tanya, you have raised your hand. Any question?
Yes, sir. Sir, जहाँ से for loop start हो रहा है data dot columns, वहाँ पे हमने -1 दिया है indexing में. वो क्यों है?
That is
-1
different.
कैसे होता है?
That's a good question. Right. So Neel, you will explain or shall I?
Yeah, you can explain. So yeah, you can explain a lot.
Yeah.
I think.
So basically, that is basically because I have taught you R programming. This is Python programming. So in R, basically indexing starts with 1. Okay, but in Python, indexing starts with 0.
Yes, sir.
Okay.
Yes, sir. वो
Yes.
मेरे को आती है.
Second thing, like there is a second thing. Second thing is that in Python, we have the negative indexing concept as well. But in R, we don't think have the negative indexing concept. Negative means there's something else. Okay. So when we are mentioning here data.columns. So first of all, data.columns means what? What are the columns? The genes, right? But the last column is what? Clinical status. Am I right, Anya?
Yes, sir.
And do you like your last one is clinical status, but आपको क्या चाहिए? जीन चाहिए, तो clinical status को remove करोगे ना अपने फीचर से.
Okay, sir.
तो उसको remove करने में ये square bracket. I told you in R, a square bracket is always used for indexing.
Yes, sir.
Slicing, subsetting के लिए. And I told you column means होता है कहाँ से कहाँ तक आपको चाहिए, right? So before column we have not mentioned anything. It means it's starting from start. Column -1. It means it will go till last, but it will remove the last column. Got it?
So this column is removed, clinical status.
Yes. So yes. So clinical status column will be removed basically. Now is it clear?
And
Yeah?
it actually, the AI gave a comment as well, exclude the last column. So
Achha,
it was
okay,
quite
we
helpful.
can answer, right? Exclude the last column clinical status. Yeah.
Okay, let's see
Any
if
doubt,
this
Anya?
works.
Yes, sir.
Chalo, then we should run code.
Yeah,
Oh,
this time,
so, but there
I
is
think you have a lot of NaN values.
yes. So NaN, there is some. We should think
Wait,
now
yeah.
that why NaN is there. Okay. Group 2 dot mean. Okay. यहाँ पे क्या किया हमने? Where is the code?
अच्छा, एक second. ये log 2
Oh,
of
sorry,
group 2
sorry.
mean minus group 1 mean होता है ना?
Right. ये वो हट जाएगा. NP dot log 2 जो पहले वो हट जाएगा. Sorry. Yes, वो उसकी जरूरत नहीं है. क्योंकि डबल लॉग हो गया तो फिर दिक्कत जाएगी.
एक second. ये correct था, I think.
Yes, ये correct है हमारा bro.
So log 4 is group 2 mean minus group 1 mean ना? Okay.
Right.
NP dot ये भी correct है, तो मैं log 2 को हटा देता हूँ.
हाँ.
So this is the same thing.
Yes.
So yeah, we just subtracting group 2 mean minus group 1 mean, and we don't need to do log 2 again because the data is already normalized, I think, as Alok mentioned.
Right. So let us see what is the result now.
Yes.
Great. Yes. So one question here, quick question for you guys. Harshin, can you explain now what is the meaning of log fold change? I explained in the last class in detail.
Yes, sir. What I can recall, it is used for the having an equal having a distribution. As if we don't do, we don't have a distribution. Like after doing log 2, we will having values like suppose -4 to +4, which will give more distributed to visualize it more easily.
That is you are telling why you are taking log. Okay, that you are telling basically like why log 2 fold change is taken. I'm asking simply what is the meaning of fold change. You are right, definitely you are right. The log makes the distribution symmetric. Okay, but I'm asking you like what is the meaning of fold change? Did you get the question basically like Harshin?
Yes, sir.
Yeah. So what is the meaning of fold change? Okay, fine. Anyone else would like to try? What is the meaning of fold change, guys?
Like there will be negative and positive values. So to make it symmetrical,
right, definitely. So what is the meaning of positive value?
If it is log greater than 1.
नहीं वो तो ठीक है, but positive का मतलब क्या है? Suppose some value is 2. What does that mean? And some value is -2. What does that mean? Okay, Payal, would you like to try? Hello.
Yes, sir.
हाँ जी बताओ, आप try करोगे? Like, what is the meaning of +2 in the positive direction?
Sir, change estimation.
Right, you are going the right direction. The change estimation, but what?
Upregulated genes.
Right, now you are right. That is, yes. Right, so positive
Okay.
means upregulated, negative means downregulated.
Downregulated.
Right, and what is the meaning of fold change? Where fold मतलब क्या होता है? Times. If you are adding A upon B, it means A upon B is 3 मान लो. So A is equal to 3B. It means A is 3 times of B. Okay, so fold change basically represents like in comparison to the given gene, how many times in the other condition it is expressed. Okay, Payal?
Okay, sir.
So yeah, so this is the meaning of fold change. So you should know the concept also behind that. Okay, why are you doing something? Yeah. Okay, fine. So last question to you guys. Yeah, sorry. So after finding the log fold change, our next term क्या होना चाहिए? We need to visualize the data as well, right? Agree, guys?
Yes, sir. So plots, volcano plots or UMAP.
चलो good है, मतलब आप थोड़ा सुनते हो हमारी क्लासेस को. चलो good. Yeah, Neel, we can go.
Yeah, so that's what we are going to do next. So we'll do the volcano plots. Again, we put the instruction here, the prompt here, design a Python function to build, I don't like this word, to create a volcano plot with data frame with genes. So we have defining what the inputs are. So inputs to this particular task would be the output from this particular task where we calculated the log fold change. And we define what differentially expressed means. So we want to do a lot of annotation as well in the plot. So we want to show upregulated and downregulated genes in different colors, upregulated in red and downregulated in blue. And we also added do not forget to show all the genes. So let's see if we can get this plot out by generative AI. Okay, volcano plot. It uses matplotlib. Yeah, okay, it does something differentially expressed. Colors, upregulated, downregulated. Okay, I think it seems good. What do you think, Alok? The answer will be correct?
Most probably, yes. Most probably आना चाहिए, right?
Okay, let's see. Oh, we haven't. We need to plot it. Wait, we need to call this function. So this we need to.
Volcano underscore plot.
And here we need to define what the data is. And this data was simply this result data frame. Let's make the name a little bit, because there are too many results.
Right.
Too many results. So we just create some LFC results. LFC. Just execute this quickly. And I'll put this here, results as which is the data frame containing the log fold change calculations. Let's see. So here the error is the data results LFC. It doesn't have the LFC column. So let's see. So it actually has log fold change. You see, the name of the column is log fold change. So we can either change in the code itself, by replacing log fold change here. Sorry. So let's see. Oh no, we need to do here as well. Okay. So I just changed the name LFC to log fold change, and there you can see this nice volcano plot with all the non-differentially expressed genes as well as the differentially expressed genes, the upregulated in I think red, right? Upregulated in red and the blue, downregulated in blue. So any questions you have?
No, sir.
Yeah, guys, any questions?
No, sir.
चलो गुड समझ में आ रहा है ना कि क्या देख रहे हैं ये वोल्केनो प्लॉट क्यों देख रहे हैं डू यू नो
Yes, sir.
चलो गुड तो बताओ आप मुझे बेसिकली बताना है कि इस डेटा से अगर हमें डिफरेंशियल एक्सप्रेस जीन को सेलेक्ट करना है तो कौन से जीन उठाएंगे
Sir, red वाले.
रेड वाले खाली
नहीं सर, blue भी.
ब्लू भी यस क्योंकि भैया पॉजिटिव हो चाहे नेगेटिव आपके काम का तो है ही
Sir.
है ना बट देयर इज वन एरर इन द कोड एरर क्या है कि इसने भैया खाली रेड वाले को डिफरेंशियल एक्सप्रेस बोल दिया है उसको ब्लू वाले
Yes,
को बोलना चाहिए था राइट नील
Yes, sir.
Yeah, exactly. हाँ.
ठीक है तो हम इसलिए कोड थोड़ा चेंज करते हैं लेट अस डू लिटिल बिट चेंज तो स्टूडेंट विल आल्सो फाइंड लिटिल इंटरेस्टिंग लाइक क्या करना चाहिए राइट तो क्या कर सकते हैं यहाँ पे थोड़ा सा चेंज की कि रेड हम लाइक फाइन सो दैट विल लिटिल बिट कॉम्प्लेक्स गाइस एंड आई थिंक आपको जब प्रोग्रामिंग थोड़ी ज्यादा हो जाएगी तो यू विल नॉट एबल टू अंडरस्टैंड सी सो व्हाट यू कैन डू बेसिकली हियर इज दैट यू कैन मेंशन इट टोटल 3 डिफरेंट कंडीशंस दैट कि अगर फोल्ड चेंज 1 से ग्रेटर वाले हैं तो वी शुड गिव यूजिंग रेड फोल्ड चेंज 1 से नीचे है तो ब्लू अदरवाइज ग्रे तो हमें कोड में देखना पड़ेगा उसने ऐसा कोड जनरेट किया था कि नहीं किया था सो लेट अस सी नील so colors red if LFC greater than 0 else blue so LFC diff exp genes log fold change okay plt dot scatter data
Oh, it's greater than 0 क्या okay हाँ
तो वो तो ठीक है इसमें ये वो तो ठीक है डेटा log fold change ठीक है -1 ये क्या इसने -1 क्यों किया -1 into data p value okay so okay fine so got it Neel so what we can introduce here changes but that will require a little bit more coding here okay for that I think we can introduce that in the code prompt I guess and we can try to generate one more prompt with that yeah that is हमें क्या करना पड़ेगा ना कि like when it is specifying the conditions here colors red fine so Neel can we do one thing here
we can show the other code which was
yes हम वहाँ देखते पहले हमने प्लॉट में इंट्रोड्यूस किया था कि नहीं किया था
so what was the other so here we define all the 3 see we actually the other code it generated all the 3 scatter plots first the non differentially expressed and then upregulated and the downregulated. So it essentially
Yes.
it created 3 plots, but here I think it created 2 plots. So we
Right.
can do the same exercise here. So
Absolutely.
maybe we
डायरेक्टली
we
कॉपी
can
से
try.
कंप्लीट कोड पूरे कॉपी कर लेते हैं. नहीं फिर लेकिन फोल्ड चेंज फिर चेंज करना पड़ेगा बार बार. लॉग फोल्ड चेंज. ठीक है फाइन.
Do you
ऊपर change
remember
करना
the code
पड़ेगा.
to rename column names?
यस नील ऊपर चेंज करना पड़ेगा. LFC की जगह लॉग फोल्ड चेंज.
हाँ, but do you remember the code? We can change the we can do this log fold thing.
Yes.
Yeah, yes, I think it is data frame. Let's see.
नहीं.
Okay, there's a log fold change something. Yeah, I have need to
चेक कर
replace.
लेते हैं कॉपी. यस.
Okay.
Great.
So now you see. So now we actually plotted 3 scatter plots: one for the upregulated non-differential expressor and the downregulated.
Sudhamri, you have this question?
I just want to ask, why are we taking log base 10?
Nice question. लॉग बेस इसलिए ले रहे हैं बिकॉज जो आपकी पी वैल्यूज है वो आपकी बहुत ही कम है. 1e - 5, 1e - 10, 1e - 50. तो 1e - 50 इज अप्रॉक्स 0. ठीक है. तो उतनी कम रेंज की वैल्यू आप दिखा नहीं सकते. तो फॉर दैट रीजन वी टेक द लॉग. When we take the negative, and it is not log 10 p value, it is minus log 10 p value. So if it is 1e - 50, so minus plus, so 50 log 10, and log 10 is 1, so it will become 50. So when it is 5, it means 1e - 5 basically. When it is 4, it means 1e - 4. Got it, Sudhamri?
Okay, thank you.
So this is for that reason, yeah.
Karthik,
Nice.
you have a question. Why are we using the log function? I think it's the same reason which Alok just mentioned. Because some numbers are very small, and then you need to.
Right.
So for the smaller numbers, we are using negative log.
Yes, because I told you in the day before yesterday class, that is like whenever you take the log, if a value is greater than 1, then the log will be basically positive. Okay, but if the value is between 0 and 1, then it will be negative value. Okay, Neel, we can show one example for that. If you say, then we can show like np नहीं एक separate cell create करके जैसे हम show कर सकते हैं np.log1 कितना है, np.log2 कितना आएगा, एक new cell create करके.
So I remove this log value, no?
नहीं ये तो मैं ठीक है, मैं ये कह रहा हूँ हमें नेक्स्ट सेल पे बस हमें एग्जांपल शो कर देते हैं कि np.log 0.1 कितना होता है, np.log 10 कितना होता है.
अच्छा,
They will
okay,
get
you
to
want
know.
to show.
Yes.
np.log
0.005 कर दो, 0.005. हाँ, yes.
So this will be very small number, -5.
I'll see. Yes, but it is negative direction basically, guys. Okay, so it is in the negative direction because it is between 0 and 1. अब एक काम करते हैं, अब हम इसे 5 कर देते हैं. So now see guys, now it is positive. So hope this answers the question now, Karthik and Sudhamri, right?
Yes, okay.
Yes,
We can show one more example, Neel. Just write np.log 1e - 50. np.log 1e - 50. -50. Yes, it will be 50 basically. Oh no.
Right.
नहीं. ये तो दे रहा है.
ये log base e पे है, I think.
10. Yes, हमें base 10 देना पड़ेगा ना. Yes, that is the reason. 1e.
अच्छा, 1e, yeah.
Yes.
हाँ.
No. और negative minus दे देंगे पहले. So negative will make it positive. So now is it clear to you, Sudhamri?
Yeah, okay.
ये P value में कुछ भी आ सकता है.
Basically,
Yes.
to identify the points in the graph.
Right. Yeah. Another question, guys? Fine. So then we can go to the next.
Yeah, another reason is when you are just just calculating the differences between these log4 numbers. It's always easier to put on a log scale because then it transforms these numbers from this very like a very exponential scale, and it puts it so that you can visualize all the regions in one data frame. Otherwise, you cannot plot everything in a single plot. So that is why you do a log transform here. so that you can see all the things in a single plot. Otherwise, the scales become too difficult to visualize all these points.
Absolutely, that is the only reason. Yes.
Okay, so now we want to filter the differentially expressed genes. Shvetandra, you had a question?
Sorry, I forgot to lower the hand.
Okay. So now what we want to do is filter the differentially expressed genes. Maybe Alok, you can explain why we want to do this?
Right. So guys, see, like अभी तक जो हमने differential expression किया, we found the genes like the blue color and the red color dots. Right. Now we're interested in what? Because yesterday, like day before yesterday, I showed you that we are not at all interested in the genes. We are always interested in the pathways. With genes, मिल रहे कौन से pathways में enriched है, right? Like these genes are coming from the glycolysis pathway. These genes are coming from cell division. These genes are coming from the brain cancer kind of the pathways. So from where these genes are enriched in, right? And for that reason, we're interested in the differentially expressed set of the genes because we know like majority of the genes are housekeeping genes in the body. And what do we mean by housekeeping genes? The genes which are mandatory for the normal functionality and the metabolism of the body, right? So we're not, and for that reason only we got maximum as the gray dots. They're not differentially expressed, right? So now we'll be filtering out these set of the genes, which are red and the blue color dots. And then we will see that upon the basis of only these genes, how well we are able to classify the patient in the different classes. For that, we'll perform the PC analysis. And after that, we'll see the enrichment that I was explaining. Yeah. Okay. So is the like the aim clear? Like why are we filtering the genes? We're just filtering the genes so that on the basis of these set of the genes, we'll perform the PCA analysis. What is PCA? I explained in the last class yesterday, tomorrow, detail. That is, because abhi 100 gene आएंगे. अब 100 gene को visualize करना it is impossible. So we'll just reduce the dimension and then visualize it. Okay. Yeah. So yeah, we are now ready to go.
Alok, can you repeat, like we have already filtered them in upregulated and downregulated. So why are we?
filter कर दिया नहीं हमने filter किया कहाँ है अभी वो तो आपको उससे plot में use हो गया ना अभी हमने result output नहीं लिया था हमने right
Okay.
Yeah, this was
Okay, fine.
it was up to your hand अगर आप चाहते तो वहाँ पे एक function create मतलब आप output करा सकते थे it was up to in your hand but हमने किया नहीं तो मुझे एक और step में करके देख लेते हैं क्या दिक्कत है right
Okay.
great
So essentially here you are doing the same thing. So here you just plotted it, but we just want to save all this upregulated, downregulated into different objects. That is the only difference. Okay. It has given. So again, it's always giving me log LFC, but I will need to like replace this because I know.
right.
Sorry, I need to also call this. And data frame is equal to. Results LFC. Result LFC. Sorry, no, it's without an S. Okay, there you go.
Great. Awesome. So see guys, so these are those genes which were enriched, like which we obtained as a differentially expressed. Okay. But anyone here guys, but कभी कभी आपने पढ़े होंगे genes वगैरह तो right because you all are bio background students. So these are the different kind of the genes. Okay, and these are basically like HUGO gene symbol names. So there are different nomenclature methods for the genes, and this is one of the HUGO gene nomenclature method that we are following here. Okay, these are the gene symbols basically. They are the gene symbols that we are having here. अब हमारे यहाँ से real journey start होती है. Till here it was all like basically the data analysis kind of thing. But here a biologist will start. कि भैया अब ये जो genes आ रही हैं, ये genes हमारे किस pathway से आ रहे हैं, PCA कैसा है इसका नहीं है. So we will see now that अब ये genes हमारे कैसे दिखते हैं.
So these
patient, yes, हमारे patient कैसे दिखेंगे इन genes के basis पे.
These includes down and upregulated, right?
Both. Yep.
Okay.
Yep. Yeah.
So now we will do the PCA analysis. So in this case, we will. Okay. So here, essentially, we have designed a prompt which will take the gene expression data set as well as also the list that we generated containing differential expressed genes as inputs. And then we want to filter out the columns in the data frame using these features. So that means we're only selecting those particular gene expression which is defined in this list. And then what we will do is now using that data frame of filtered genes, we will do the PC analysis and try to plot the PC1 and PC2, the first two principal components. And hopefully, and we'll also color these samples as per the categorical variable. Okay, go ahead, Tanya.
So what is Seaborn?
Seaborn is a Python package it's a Python package to visualize. It's slightly it's slightly better than Matplotlib. Just let's just say that it produces better graphs, and the amount of code that you need to plot using Seaborn is slightly lesser than the amount of lines you need to write when you're using the Matplotlib library, which is one of the most standard libraries. So that's why we prefer Seaborn if you want to produce publication quality plots in Python. So let's see. Okay, so we now generated the code, and you can see here we use the Seaborn package here, and also we will be using the generative AI imported the PCA function from the sklearn.decomposition library. And you can see, let's go step by step what has happened. So it takes the features and data frames. It filters the data based on these features, calculates the PCA, the two components, and then and creates the principal component data frame. And then and the last step, it plots both the principal components. It also colors here, I think, yeah. It also adds the last column of the original data frame in this principal component data frame containing these first two principal components. So essentially, we are having a data frame with 3 columns. One, the first two principal components, and the third column is essentially to color the genes based on the clinical condition. So I will now see if I can plot this. So for this, I will I'll call the function. So features here are my this list of features which we already calculated. Oh, we did not save it somewhere. Let's just say, diff express gene list. I'll just save this in a variable, and then I can call here, and then the data frame here is my gene express data. Let's see what happens. Gene express data is not.
Neel, name of our data GeneXP only.
GeneXP, sorry.
Right.
Okay, somewhere something has gone wrong. Let's see.
Okay, so got it. यहाँ ऊपर आते हैं. I've got the error. Error क्या है? बस यहाँ पे देखते हैं एक second. Filter data frame is data frame features. So we mentioned GeneXP. Okay, PC underscore 1 and 2. PC dot fit underscore transform filter underscore data principal underscore df pd dot data frame data principal components column is final.
I think here.
हाँ नील एरर ये बेसिकली वो आपका इंडेक्सिंग एरर होगा मोस्ट प्रोबेबली है, क्योंकि जो प्रिंसिपल कंपोनेंट जनरेट होके आ रहे हैं वो तो आपका वो होगा 0 1 2 आईडी रखा हुआ इसने, पर जो हमने डेटा फ्रेम से वहां से जो कॉल किया है लास्ट कॉलम तो उसकी इंडेक्सिंग कुछ और है, तो इसलिए वो एरर आ रहे थे. काम कर सकते हैं इसको, जो हम यहाँ पे कॉन्केट कर रहे हैं डेटा फ्रेम डॉट ILO से इसको लिस्ट बना देते हैं. ठीक है. Or we can do one thing here. We can make a change here in the code. That is principal underscore df. We can add the last column is clinical underscore status and the name of the data frame. Third step, yes, यहाँ पे इसके ऊपर. Yes, यहाँ पे we can write this. We need to write this. नहीं, उसके नीचे. Yes, here. We will write this. Principal underscore df. स्पेलिंग कॉपी पेस्ट कर लेते हैं ना, जो लिखा हुआ कॉपी पेस्ट कर लेते हैं ना फोटो को. राइट, स्क्वायर ब्रैकेट क्लिनिकल अंडरस्कोर स्टेटस.
आई डोंट थिंक इस पे क्लिनिकल अंडरस्कोर स्टेटस आया होगा.
नहीं हम ऐड कर रहे हैं अलग से कॉलम, हम कॉलम नेम ऐड करेंगे यहाँ पे बेसिकली. यस, क्लिनिकल स्टेटस यस इक्वल टू, एंड जो नीचे उसने कॉपी किया ना उसको पेस्ट कर देते हैं डेटा फ्रेम डॉट ILOC वाला. नहीं हाँ ये नहीं ये भी चलेगा. यस, ये भी चलेगा. और इसको लिस्ट बना दो, इसको लिस्ट इट विल रिक्वायर टू गुड लिस्ट टू लिस्ट. ओके फाइन, ग्रेट. एंड नाउ नीचे वाला कोड हट जाएगा हमारा.
अच्छा, यू आर सेइंग दिस वन शुड बी डन.
यस, ये पूरा लिस्ट हो जाएगा. यस एंड नीचे वाला कोड हट जाएगा, उसकी जरूरत नहीं हमारा फाइनल DF की. नहीं नहीं ये नहीं, ये नहीं जो फाइनल डीएफ कमेंट कर देते हैं. यस एंड नाउ प्रिंसिपल डीएफ यू इज इक्वल टू में चला जाएगा आपका लास्ट कॉलम नेम, स्टेटस नेम ओनली. यू इक्वल टू स्टेटस लिख दो, खाली क्लिनिकल, ओनली क्लिनिकल स्टेटस. Just write clinical status, it will take. Yes, clinical status. Now it should work basically, what I think. Let us see. Great.
Yeah.
Yes. So great. So now here we are able to get the sample. So guys, now you can see now, like the chat GPT will give you the wrong answers, or basically the AI will give you wrong answers also. But for that you need to have good understanding of programming. कि भी आपको थोड़ा बहुत मैनुअली खुद चेंज करना पड़ेगा, ठीक है. वो भी हो जाता है चैट जीपीटी से, उसको फिर इटरेशन देना पड़ेगा अगेन एंड अगेन एंड अगेन. इट विल जनरेट, बट उसमें टाइम लगता है. राइट नील?
Yes, yes. So I think I'll answer Shwetam's question. What are PC1 and PC2? So अगर आपने देखा होगा हमने जो the original data set. So you see there are many different genes here for each of these two classes. So what essentially we did using PCA is we reduce all these columns into two main components. So principal components are think of it dimensions where this data can be reduced to. More technically, where these are the two directions where the variance of the data is maximum. So essentially, we reduce this entire data set into a set of principal components. So usually PC1, PC2, PC3, PC4, PC5. But mostly we are interested in the first two principal components because the first two principal components often describes the whole of the data. So in a nutshell, you can compress this whole data set into two principal components. And that is what we are doing. So, and the reason we are doing is to visualize the underlying distribution of this data set, to see if there are some interesting patterns, because you cannot plot all these genes in multiple dimensions because it's impossible to visualize. So the best way is to reduce all these the columns into two columns and then visualize it into a 2D plot, and which is actually what PC1 and PC2 are. And then we are then simply coloring PC1 and PC2. So instead of like all these genes, now we have PC1 and PC2. Just imagine there is only PC1, PC2, and then we are just coloring based on the clinical status here. And that is what happening in this data frame. Understood?
Okay.
Hello, sir. During PCA, the principal component analysis, how many features, how many PCA components we can generate for a data? Like, is equal to the number of features in the data?
Yes, usually it's the number of features, but there's a plot called the principal component variance plot. So some of the variances.
Scree plot is the name. Scree.
Yes, Scree plot. What's it called?
Scree. SCREE.
Okay, Scree plot. Maybe we can show this, no?
Sure. Because
I don't remember the exact code, but
उसके लिए हमें वो करना पड़ेगा. मैं बताता हूँ. Like, do one thing, Neel. So for that, we can just copy paste. वो हम manually करके दिखा देते हैं. Right?
Okay.
So we can just So we can नहीं हम copy कर लेते हैं कोड ऊपर वाला PCA वाला. PCA वाला कोड copy कर लेते हैं. पहले first line हो. Yes. हाँ बस. Yes. And yes. अब यहाँ पे हमें चाहिए PCA. PCA is so we need to copy only that particular component, the first and the second line, and the rest is not required. The third, first, second, third is required. Neel. First, second, third is required. Yes. First, second, third line is required. बाकी सारा हटा देते हैं. बाकी सारा हटा देते हैं. Yes. नीचे वाला भी, because we will do it manually, so no function is required here.
Scree plot, no? What did you say?
नहीं वो फंक्शन नहीं बनाया. नहीं पहले मैनुअली करके दिखा देते हैं. We will put it in a function.
अच्छा, okay.
So for that reason I was saying only we can select the first three lines and then we can start the coding.
सर
हाँ जी तान्या बताओ.
सर जो पीसी एनालिसिस में डायमेंशन रिडक्शन होता है उसके लिए हम अपनी कंडीशन देते हैं, मतलब जो भी हमारा कोड है या सिस्टम है उसको हम कंडीशन देते हैं कि हमें जो रिडक्शन चाहिए वो सिमिलैरिटीज के बेसिस पे चाहिए या फिर डिसिमिलैरिटीज हमने जो जीन दिए हैं उनके बीच में, या फिर वो अपने आप ही खुद ही कुछ भी करते हैं.
So Neel, you would like to take this question?
Okay, can you come again, Tanya? Sorry, मैंने सुना नहीं था.
सर पीसी एनालिसिस में जो डायमेंशन रिडक्शन होता है, हमारे पास जैसे काफी सारे जीन्स हैं इस वाले एग्जांपल में भी, और हमने उसको दो कंपोनेंट में रिड्यूस किया है, तो वो किस बेसिस पे होता है? हम खुद डिफाइन करते हैं या फिर वो सिस्टम अपने आप कर लेता है?
तो नहीं, तो usually we reduce it. So actually PCA analysis is reducing it to as many columns you see. So those many principal components exist. So, but we only choose the first two principal components because it explains the most of the data. So that is why the reason we are choosing PC1 and PC2, because it explains the almost all the variance in this data set. So, and that is why हमने define किया कि हमें first two components ही plot करो. अगर if you put 3, तो you will get 3 PC components. If you for 8, you will get 8 components. But it doesn't make sense to plot all the 8 components. So we just want to plot the first two components. So usually हम जब 2 डालते हैं, तो you get the first two components by default in this.
Okay, sir.
Yes, so you want to ask something?
Yes, Alok, in this syntax, we have not like defined that the clinical status will be defined as blue and orange color. So can you help me that how have we done this in this graph?
नहीं वो मैनुअली
Can you
कर रहा है,
come-
वो यू पास किया ना, यू कॉलम से ले रहा है वो, हमने यू पैरामीटर दिया है.
U clinical.
Can you-
Okay.
Can you come define your question again?
I'm asking, like in the graph, it has been mentioned the clinical status is GNG is shown as blue dots and MBL is shown as orange dots. So, in the code is it written?
So here I have defined here the clinical status, I think. So here I have defined hue as clinical status. So in this scatter plot function, I define the data frame, the data containing the principal components, and I also ask it to color it, which is here I give the argument hue and to put clinical status. So essentially it's coloring based on the clinical status GNG and MBL. Did you get the answer?
No, I still don't get it.
You were asking how it got the colors.
Yes.
Okay, so
You
let me
का
show
पूरी
you
basis
then
पे है.
step by step. Wait. So I will also, what I'll do is, ये जो principal DF है, so here I calculated the two principal components using the original data set. Then I added a column in the principal DF. So I'll show you what this data set is, what this new data set is containing the two principal components. So let me see. Principal DF, and I need to print it out. So here you see, I'm just printing the first 5 rows of this principal component data set. So in this data set, it contains the first principal component, the second principal component, and also the clinical status, which is actually GNG, because I put, but I can show you if you put sample, which I'm saying like say 10. So you can also see some MBS as well. So, and then when I'm plotting, then what I'm essentially doing is I'm plotting this whole data frame as a scatter plot. So what I'm taking as an input is the data, which is principal DF here. Then I define which two columns need to be plotted, principal 1 and principal 2. And hue is a way of saying, okay, I need to color these scatter
Hue
points.
is the function. I think hue is the function that is assigning the colors to the points.
Exactly.
If there is
exactly.
X and
Hue
Y,
is-
it is automatically assigning blue and yellow. If it would be X, Y, Z, it would be assigning 3 different colors. So hue is the reason that is assigning colors to the scattered
Exactly.
points.
Exactly.
Yeah.
You are right. Hue is the argument which tells, okay, color this to color these scatter points based on this particular information, hue. And hue, I have defined here what this information is, what this column should be. This is the clinical status column of this data frame, which you can see here.
Okay. Okay, fine.
Okay. I also want to show you the variance problem. So let me see if I can get this running. Let me get this PCA variance on why do we select 2 columns. So principal data frame here. I'm just opening up the documentation to plot this PCA. Okay. Let me see if I can, say get this data. But this time we'll not use the just to quickly save some time. So let me see PCA virtual components. PCA. Let me see. Let me just put say 8 components. Okay.
We can just like Neel, we can just leave that column and then it takes all the possible components that will be generated.
Okay. Yeah.
We don't need to mention anything here. Yes. We don't
Yeah.
need
Let's
to mention anything.
put,
Yes.
yeah. PCA full. Let me put this like this, and let's see if I can generate this. Okay, now you see what essentially we did was this Cree plot. I hope I'm getting the name correct. So it has, I think this data frame had, I think 35 somewhat genes. So we generated
no. Yeah, so there is basically like one that is proper statistics.
N-1 components we should have. N-1, 34. I guess.
Yes.
I'm not sure what is the number of columns in this data frame.
Yes, no, not
but essentially.
Neel, basically here it goes into the proper like maths for that. So it takes whatever, like if the number of samples is less compared to number of features, that it builds the PC for the sample cross sample. Okay, because in our data we have 34 features or samples. So that's why it has generated 34 PCs. So the reason is for that. Okay.
Yeah, so and why we chose the first 2 components, as you can see, the explained variance in this data set, which is the spread. You can think of the data set as a spread. It's most explained by the first 2 components. So if you sum the first 2 components, you get the maximum variance explained in this data set. And that is why a common practice is to plot the first 2 components in this PCA plot.
Can you tell me in this graph, where can we see the first component, like from 0 to 10? Do you want to say?
So this is component number 1. This is component number 2, component 3, component 4, component 5. That's how. Principal component. So you can see on the x-axis. So x-axis it represents the component number 1, component number 2. So this is a bar graph. This is not a
first 2 bars referring to PC1 and PC2.
Yeah. So we are essentially plotting PC1 and PC2.
Okay.
Cool. Any other questions?
No, sir.
Okay, that was a good discussion. So now I think we'll, Alok, do you have any other sessions planned or?
We can explain this so that they will have a complete idea for this.
Okay, cool. So we now go to enrichment analysis. So here, I think, Alok, you can explain what this analysis is.
Fine. So as I told you guys, like a day before yesterday also I was explaining you what is the meaning of enrichment. So basically like we have obtained a set of the genes, but now we really want to understand these things at the pathway level, right? And the genes that we have got, these are enriched with which sort of the pathways. Like they're coming from the cancer pathway, they're coming from the HIV, they're coming from some drug-related pathways, or from any particular specific condition. They are related to some biological process or some metabolic process or something like that. Okay. So for those reasons, we are interested to find the enrichment. Okay. कि भैया ये genes आपकी किस pathway से आ रहे हैं? That means enrichment analysis. Okay. And for that, we have a large number of different databases. So Neel, if you allow, can I share a screen for 1 minute?
Yeah, sure. Go ahead.
So great. Let me know, Neel, when the screen is visible. Hi, Neel, is the screen visible? Great.
Yes.
So, guys, yeah. So there is a site called MSigDB. Okay. This stands for Molecular Signatures Database. And then when we go here, you will find something like this here. Human collections, right? Hallmark gene sets, positional gene set, curated gene set, regulated target gene set, computational gene set, cell type signature gene set, immunologic signature gene set, oncogenic signature gene sets, and all. Now suppose I just click upon oncogenic. Chalo, इस पे click करते हैं एक बार. Let us click upon this, and now you can see here that 5219 gene sets, immunologic signature gene sets. It means what? There are 5,219 gene sets. One set will comprise the number of the genes. Suppose if you want to click upon this, now see. So these are the name of the set of the genes, like gene sets basically. And now suppose if I'm going to click upon, किस पे click करें? Positive thymocyte. चलो इस पे click करते हैं थोड़ा सा. Let us click upon this. And now when we click upon this, we have got something like this here. Now see, standard name, this systematic name, genes upregulated in thymocyte. So this is the description of this particular gene set. That is the genes upregulated in thymocytes. What is thymocyte? You must be knowing. So basically, it is the organ in the body where the T cells are being generated. Thymus. Okay, so thymus is the organ where thymocytes are being produced, and then T cells develop. So it is the description in the detail. In collection, then you will go bottom now, related gene sets. So 19 additional gene sets. So 2 gene sets from the same authors. Now download gene sets. So if I'm going to click upon XML, TSB karte hain, theek hai? So let us click upon TSB. Let us see in what format is going to create. Great. So I will now just copy paste this. I will delete. Now, sorry. Yeah. Now I will go to our workshop. Now I will create one Excel file here. Now see guys, okay. So what I want to show you is that this only, okay, gene symbols. Okay, so see, these are the genes. Now you can see now that these are the genes which are basically enriched in this particular pathway, which we have collected. Okay, so now what is the meaning of enrichment analysis? That you perform differential expression analysis, you got some genes. Now you will look at the intersection of those genes with these set of the genes which are already predefined. From where did we get these predefined sets? By the different scientists from the different research labs and all. Okay, so there are so many mathematicians, there are so many wet lab scientists and all. So they perform the experiments, they find the different set of the genes enriched in different set of the pathways, and then a large number of databases are there right now, nowadays. You perform DEA, you got the set of the genes. Now we basically look at the intersection of this our gene set with this particular gene set intersection, and then we just try to understand that. Suppose you got total 35 genes, and this gene set contains total 500 genes. So from the 500, and you have the 35, how many genes is there in the intersection of both? And that will be basically represented then again by p-value. Like either some statistical test is being involved here. Which one? That is your hypergeometric distribution. So based upon that, we get some p-value, and that really signifies how significant these genes are there in this particular pathway. Okay, and this is the real meaning of enrichment. Like this, we have thousands of pathways, and that is being done there, analysis, and then we get the set of the pathways as the enriched. Is it clear to all of you guys, the meaning of enrichment analysis or not? Please let me know. ये कुछ तो बोल दो, yes or no?
Yes, yes.
Chalo, good. Okay. So this is the meaning of enrichment analysis. That is what now we are going to see. Okay, because trust me, biology is not that simple. Okay. Stats, maths सब अपनी जगह, but biology में आपको सबको एक साथ मिलाना पड़ता है. So that is not that simple. Okay. So yeah. So over to you, Neel, now.
Okay, thanks for the explanation, Alok. It was good to revise the concept again. So now we will do the enrichment analysis using the GCP package. I think this was a package originally meant for R, but I think that they have also released it for Python. The input should be differentially expressed genes. So let's see what we get.
Great.
Okay, differentially expressed genes. So we should here pass the that list, right, Alok?
Right. The filter differential express list.
Yes.
And we can save this result as well, Neel, in some variable. We can write it in this pathway, ENR path. Yeah. Great. It might give error, I think. Because GSCA is not installed, so it could give error.
Oh yeah, we did not import this, no? GSCA.
नहीं वो आपका import हो रहा है, वो कोड में import हो रहा था. ओ आ गया result, छोड़ो. Okay, fine. So it is basically working on the नहीं आ गया result Neel.
वो तेरा दिमाग चलाना पड़ता है. कोई कहीं से बोला, कोई कहीं से बोला, मैं तो उससे सारा.
अच्छा Neel, just a quick question I want to know from you. Hello.
Yeah. Yeah.
So a quick question that I wanted to know basically. So when we are working on this Jupyter notebook, which is working on somewhere the cloud, so here we do not need to install the packages, right? Is it so?
दो बोल रहे हैं और एक अंदर से.
We need to install.
Can you mute yourself? Whosoever is there, can you mute yourself? Yeah, thanks.
नहीं हो पाएगा.
Okay. So what I was saying was, if you're working on Google Notebook, Google Colab, then you only need to install the packages here. Like all the other packages we are usually installed from by default, but there are some additional libraries that you need to install. For example, GetFast, Jupyter, and GCP, for example.
Okay.
So I think this analysis is still going on. Let's see. काफी time लगा रहा है.
Normally it should not take that much time.
I think this organism, this is correct, right? Check. We have
Yes,
the alternate.
Human is
This is
fine.
okay. This is another code. This gene set, it took like gene. Maybe we use this code because this code I'm not sure how much time will it take.
Like both should work. There is just we can just copy paste that as well. We can see if it works.
Okay, let's copy paste this.
But we'll have to terminate that now, right?
This one is kind of
great. Terminate हो गया. Fine. यहाँ पे अपने लिस्ट पास करना पड़ेगा नील only yes.
चलो, let's see.
Great. Fine. So see guys, now here is the result in your hands now. Okay. So you can see here that we passed a path like gene set here. We have specified only one gene set here. For that reason only you will see on the gene set only one process, right? There are so many packages right like this. So this right now the example it is giving you the way I gave you one example. Okay. So the gene biology, GEO means stands for gene ontology, biological process 2018. Term, so it is basically the name of the pathway. So basically the gene set will contain a large number of set of the pathways. Okay. It means what? कि भैया biological gene ontology biological process जो 2018 में version release किया गया था इसका मतलब ये है only. Term means it is the name of the pathway. Okay. You see regulation of upregulated secretory pathways, organelle membrane, modulation of chemical synaptic. Okay, and all. Then we are having here overlap. Overlap means what? I explained you like intersection. कि भैया आपके इस वाले जीन सेट में सपोज 100 जीन्स आर देयर और आपके जो लिस्ट है उसमें कितने 100 में से मिल रहे हैं आपको राइट so that is being represented by overlap. That is 5 out of 37. It means this regulation of regulated secretory pathway, the gene set contains 37 genes. Out of that, you have got 5 genes. P-value, this is basically done by overrepresentation method. The name of the method is overrepresentation method, which is based upon hypergeometric distribution. So like the normal distribution, hypergeometric distribution is also one type of the distribution. Okay. So if any of you studied maths in class 12, so you must be understanding that Bayes theorem. So it is somewhere associated with the Bayes theorem, basically kind of the thing. So basically, hypergeometric distribution is being involved here, like applied, and then we get the p-values from here. Okay, then we have the adjusted p-values because multiple hypothesis testing correction will multiple hypothesis testing is being done here. We need to adjust the p-values for that as well. Old p-value, forget about this. Odds ratio. Odds ratio is what? P by Q. P basically means the probability of success. Q means probability of not getting the success or the failure. Okay, so it is basically given for that. But you are only interested in 2 things here. 3 things: term name, p-value, overlap, and the name of the genes. ये pathway पता चल गया और कितने significant है और कौन से gene है वो, right? So like suppose that if you are going to look at the first pathway, we are getting here how many genes? 1, 2, 3, 4, 5. SYT4, SIT L4, SIT13, SIT1, RAB3A, RPH3A. So these are the genes which you are interested in. They are the genes which you are interested. Not interested, but they are the genes which have been enriched and found as differentially expressed, which are associated with this particular pathway. Similarly, if I'm going to look at this particular pathway known as number 462, regulation of cellular macromolecular biosynthesis, we have got only one gene from 631. Okay. And the gene name is basically ZNF536. Excuse me, just a minute. Yeah, sorry guys. Yeah. So this is the gene here, ZNF536. Okay. So this is basically how you basically understand this plot. And trust me, from here onward, the real research starts. Okay. तो इतना तो जैसे हमने आपको 1.5 घंटे करके दिखा दिया, so you can also do this thing, not an issue. But the real research and the analysis starts from here onwards. अब आप इन जीन का क्या करने वाले हो? Okay. So अब आप इन जीन से क्या करोगे? So Like one analysis that with the help of these genes, we try to see now that what accuracy we are able to get in classifying the two different classes of the patients. Are we really able to classify the patient into two different classes or not? If yes, then it is great. Because in machine learning, we have the concept on the feature selection. Similarly, in biology, this is also we are doing nothing but simply feature selection. That we are simply selecting the features. Right, Neel?
That's right.
Right. Yes. So do anyone have any question here, guys? Hello? Hello? Okay, fine. So I don't think they have a question. So either they understood nothing or they understood everything. Yeah. Fine. So this is the last thing that we is like. I think we should skip this, Neel, I guess. Right? Or shall
Okay,
we?
let's skip this. Yeah.
Yes. Because machine learning, because the student will not be able to understand, so it is not beneficial as well, right?
Okay. So
Great.
so I will just quickly go through my there's some essential things that I
Absolutely.
would
Yes.
also
Sure.
like to touch. So what we just showed you is how to do your analysis using generative AI and Python notebooks. But it's also important that the research that you do, the data analysis that you do is reproducible, it's shareable. And for this, we often use Git version control system. Some of you may have already worked using Git, but for those of you who do not know how to use Git, so today I'll also give you a quick demo of what it is and how you can use it to do reproducible research. So so what Git is, it's a version control system. It can track changes in code. It can be used to track changes in data analysis scripts, experiments, and data. And it's really important to replicate all the analysis. And repositories like GitHub allow code sharing, and also you can. So these days, you would often be requested by journals or reviewers to share the code with them whenever you are submitting something for review. So it's becoming very important to know these skills as well. So in a nutshell, how it works is, for example, if you're working on the main branch of Git, and I'll show you what this main branch is. So if I go back to my Git repository, our original Git repository, so this is what is on the main branch. This is called the main branch, and in this main branch, I have already included some content. For example, I have included a README file. I have included the code book, which Alok and I co-created before even the start of this workshop, just to be sure that all the prompts and all the output, all the code was giving correct results. So we just made sure we debugged all the code, and we actually saved all the code in this particular branch, which is called the main branch here. But as you saw in today's analysis, that a lot of the code was not working, and some code we had to change manually. So definitely the code that we generated today during our workshop will be much different than the code what Alok and I had worked on and saved it on the main branch, which was done a couple of months back. So how should we then manage this particular the code book? How should we version control this particular code book? And the answer is quite simple. So what we need to do is, of course, either you use the terminal. I don't know. I'm not sure if you know how to use the terminal, but that's for advanced software engineers and for advanced data scientists. Whenever you would work in a corporate, you would have to know the skills of how to use terminals to work with Git. But today I'll just show you how you can use the web interface and how you can already get started working with software like GitHub. So to do this, what I will do now is I will save this particular workbook on my local computer, on my local drive. So we will save notebook as, so today is I think 31st demo, 31st Jan 2024. So I'll just save this on my code. Oh, sorry, I did not. Apologies. I think I should have saved this one. Oh, there are too many. This is fine, but let me just quickly check it. Just make sure. No, this is right. Let me make sure if this is this is the code book. Yeah, we did save it correct. So this is now saved on my local computer, so I can show you quickly, where this file has been saved. So you can see, so this is a local folder on my computer, and I just saved this file, workshop codebook demo 31 Gen 24. So I just saved this file. Now I will show you how we will upload. I will create a new branch on GitHub and save this codebook. So now I will go to and share again my web browser. Okay, we're back on our GitHub repository. I just described to you what this main branch was. So now I'll just create a new branch, call it and name it as demo 31st Jan. And so now what will has happened is, so I have the main branch here, and now I have the branch which was created out of the main branch, and this branch is called 31st Jan branch. So here I will add the code book that we generated today. So I will upload files, go to upload files. I can quickly drag and drop here, the same file which we did 31st. So here I'm just uploaded 31st Jan, and I will write a commit message, uploaded 31st Jan demo output, and I'll just commit these changes. So in this 31st Jan code book, in this 31st Jan demo branch, I have both the today's code book and the original code book that Alok and I had prepared. But if you go to the main branch, which was what Alok and I prepared for, what we prepared, so it doesn't contain the 31st branch, 31st Jan, it doesn't contain the code book that we created today. So this is a good way of doing version control of your analysis. Sometimes it's very helpful. For example, if you're changing certain, if you're doing certain experiments with your data analysis, and you want to share, if you're doing an analysis with different parameters and you want to save the output, if you want to save the results with these different parameters, so this is a good way of doing this. You create different branches for these different parameters, and for each of these parameters, if in each of these branches you are saving the code book generated with that particular parameter. And then you can easily, suppose I want to share the code book or the repository containing today's code book. What I simply need to do is take this link and then share it with the code reviewer or whoever would be interested in checking out today's notebook. So that's in a nutshell what Git does. It helps you track the changes in your scripts. I also want to discuss a little bit about data confidentiality. So want to highlight and want to create some awareness on how your data is being treated. I would like to emphasize that the data that you work with, it doesn't belong to you. It often belongs to your organization or your university. The data you get from the experiments, you do not own it. And as a result, you should treat this data with a lot of respect and a lot of confidentiality. You should not upload this data randomly on any server. Definitely, you should not send it to ChatGPT so that it can improve itself based on your data set. And typically, what happens is when you're using the ChatGPT web application, the ChatGPT web application uses the data that you send to update itself, to improve itself. Whereas the modality, the workflow that we showed today, we use only the APIs, not the ChatGPT web application. We use the models in a programmatic fashion. We use the APIs only, and based on OpenAI's terms and conditions, when you use APIs, they do not use that data to train or update their models. So it's a slightly more safer way to use the data in a more confidential and private way. And that's why I also highly emphasize the fact that never use the web application to perform any kind of confidential data analysis, and never upload any confidential data sets on your private cloud storage. For example, do not use your own Google Cloud or whatever cloud, or Bob Cloud, or what have you, or iCloud to upload any kind of confidential biomedical data sets. And finally, as we saw in this exercise, that just using learning generative AI tools is not enough. Of course, it can really accelerate your data analysis. It really generates a good coding template at first go, but you also saw that you had to sometimes correct the code as also there were a couple of bugs, and you need to understand the basics of coding at least to figure out the bugs in AI generated code. So that is why you would need to know some fundamentals of programming in Python or R. You would also have to know basics of biostatistics and machine learning as a result. Otherwise, you will not be able to debug the code generated by AI. I would also highly recommend to assess the accuracy of the code, relevance, and there are also sometimes biases in the code, in the output, and you need to be well aware of these issues. You should also know how to conduct analysis in a way it is reproducible, that someone is able to reproduce your entire analysis in its entirety. And for that, Git version control is a good way to do this. And yeah, of course, always be careful of how you manage your data and how you manage your code. You again do not often own all these artifacts. Your organization or your laboratory or your university does, and it may create a lot of liability if you are not careful enough when you're working with confidential biomedical data sets. So with this, we have come to an end of today's session. And Alok, if you would like to connect with me, you can scan this code and you can connect with me on LinkedIn. I also put the scan for Alok. You can also connect with him if you are interested.